Enzyme kinetics, part 3. Let's continue. So what about enzyme catalyzing reactions that involve two molecules, two units? In fact, it is often the case that you'll be having a multi-substrate reaction where this enzyme is going to take A and B, or even A, B, and C, and bring them together in such a way that you produce your product, be it D or DE or DE and F. Where we have two units or two substrates involved, we call that a bisubstrate reaction. And there are two mechanisms that we can look at for this to occur, and we can look at the kinetics of those two. We can talk about a single displacement or sequential, or we can talk about a double displacement, which is a ping-pong approach. So when we look at the single displacement reactions, they can either be random, which means that if I'm substrate A and I get to the enzyme first, then I get to react. If I'm substrate B and I get to the enzyme first, then I get to react. Or it can be ordered. That means that if substrate B gets to the enzyme first, enzyme's going to say, A always comes before B in my reaction. So in ordered, the lead substrate has to bind first. So A would have to get there and bind before B is allowed to get in. So we can either have a random single displacement reaction or an ordered single displacement reaction. So if we look at this random single displacement reaction, let's look at the details of it. We have reactant A and B getting together to form P and Q. This reaction, however, requires the enzyme E. So we have our mixture in situ, or in a living system in vivo. So we have the free enzyme represented by E. Now this enzyme in the mixture has access to A or B. In either case, whichever one hits the enzyme first, it is going to connect. So we can form the enzyme substrate complex AE or the enzyme substrate complex BE. Now, the goal is to get A and B together. The enzyme is what's going to do that. And so we form the two complexes independently, AE, EB. In order to get to the next stage, these two complexes, these two binary complexes of the enzyme plus one substrate have to get together to form the ternary unit, where we have the enzyme combined with A and B. And notice that we start out with two units of enzyme, one A and one B. When they get together, one unit of enzyme is released, and we have AEB, the complex. AEB, provided the conditions are sufficient to drive the reaction forward, they then convert to PEQ. So we have the enzyme with the products as a complex. Now remember that the goal was to release free P and free Q, and so we bring in a free enzyme unit, which is then able to pick off the P, giving us free P, the product, and enzyme, leaving behind enzyme Q complex, which then can break down to release free Q. The enzyme is now available to go back and pick up more A or B and keep the process going until we have some kind of mechanism that says, hey, stop, we have enough P and Q. And we talk about those regulatory methods later on. So this is the single displacement reaction. It's random. In other words, it doesn't matter who gets to the enzyme first. Ultimately, they form the complex with both substrates and the enzyme, and then they produce the products. Now, in an ordered single displacement reaction, we have to start out with the right substrate. So in this case, the leading substrate we designate as A. And so what happens is that in order to start the whole reaction sequence, we need A to complex with the enzyme to produce the AE complex. So that B does not combine with the free enzyme. B combines with the AE complex. Once that happens, we form the AEB complex. That complex then is going to form the product complex, which ultimately breaks down to give us our products and the free enzyme. And remember, the free enzyme can then be circulated back to pick up more A. So the difference is in the random single displacement, any of the substrates can start the process. In the ordered single displacement process, a particular substrate, that same substrate, must always be the one to start the process. This is another way of portraying the ordered single displacement reaction. Take some time, pause, have a look at it, and make sure that you understand what's going on. So when we talk about catalyzing the bimolecular reactions, what do the kinetics look like? Well, in a single displacement bisubstrate mechanism, notice that as we increase the concentration of the second substrate, we get a change in the slope. Think about why that is. Pause, write down what you think, and check out your notes afterwards. So point to note that increasing the concentration causes this change. Now we also have what we call the double displacement. In this case, you can also describe it as a ping-pong mechanism. It may seem complex, but let's simplify. So we start out with the free enzyme, which is always the case in reactions. You have to start out with the free enzyme because if the enzyme isn't there, reaction pretty much not gonna happen. We then have substrate A. Substrate A binds to the free enzyme to form the AE complex. As that happens, we get the first part of the reaction. The first part of the reaction changes A, substrate, into P, product. So we now have the product enzyme complex. The product is now released. So what has happened? We've converted A into P. But we're still trying to get Q as well. So we now have the activated free enzyme. At this point, substrate B enters and complexes. As substrate B complexes, we get EB, which undergoes the reaction to form enzyme with product Q. Product Q is released by the enzyme, and we get the free enzyme, which can then be recirculated to pick up more A. So in this case, a double displacement, you have a definite start with A, you release one product, bring in one reactant, another substrate, release the other product. So it's described as ping pong because if you know the game of ping pong, which is also like the game of tennis, you hit one side, it hits back over again. And so you bring in one substrate, you get one product, you bring in another substrate, you get the second product. So ping pong. So what do the kinetics of this type of reaction look like? That's an alternate way of showing it. Be sure to pause and have a look later on. Make sure you understand it. So in a double displacement reaction, right, by substrate, notice that as you change the concentration of the substrate, you get parallel plots. You recall, let's go back, that in the single displacement reaction, as we increase the concentration of the second substrate, we saw a change in the slope. With the double displacement, the slope remains the same, but the intercept varies. So we get parallel lines as we increase the concentration of B. Again, pause and think, what does that mean and why is it so? So we've seen that enzymes need specific conditions. We've seen that enzymes can be inhibited. We've seen that enzymes can act using single displacement reactions, using double displacement reactions, and we've seen the impact on the kinetics. Now, how can enzymes be so specific? How is it that one enzyme doesn't cause multiple reactions? Well, one hypothesis was the lock and key, that the enzyme and the particular substrate fit exactly. So that if the substrate didn't fit exactly into the lock, which was the enzyme, the reaction didn't take place. So the lock and key hypothesis sees the substrate as the key and the enzyme as the lock. Another hypothesis was found to be actually more like what was observed, and that hypothesis was the induced fit. And in that case, what they're saying is that as the substrate comes in, it modifies what is happening at the active site so that the enzyme's active site better fits the substrate. Now understand that if the enzyme and the substrate are totally unrelated, you're not going to get that happening, but the enzyme has the basic shape at the active site, and then when the substrate comes in, as it comes in, it actually causes minute changes in the active site that makes for a more perfect fit. So when we talk about specificity, we're talking about how well this particular enzyme can pick out a particular substrate. And specificity and reactivity are often linked. So that if we look, as we will later on, at the hexokinase reaction, when glucose, which is the substrate, binds at the active site of the enzyme hexokinase, it actually induces or causes a conformational change in the enzyme. And what that means is that there's an actual shift in the physical structure of the enzyme so that the hexokinase actually closes in on the substrate, making that catalytic site. And it is quite a wonder to see at the microscopic level that the enzyme actually reaches around. You can almost call it an enzyme hug. So, enzymes are specific, they need particular conditions, and they can behave in certain ways depending on how many substrate units are involved. At the beginning, we talked about the fact that enzymes are mostly protein. Does that mean that non-protein structures are unable to catalyze reactions? Actually, no. There are some ribosomes that have enzyme activity, and that is in the absence of protein. In other words, traditional enzymes are not present, and yet the ribozymes have some activity. There are also enzymes, which are antibodies which will bind the transition state of a reaction of interest, and these can actually be created or synthesized. So the answer to the question, no, not all enzymatic activity is caused by proteins. Not all enzymes, substances which catalyze biological reactions, are proteins. So then, could we actually make or design an enzyme for a particular reaction? Wouldn't it be awesome if we could design an enzyme that we take in a tablet and it automatically sucks up all the belly fat? Anyway, that's a dream. But scientists have been working and attempting to engineer or make enzymes by in vitro mutagenesis. In other words, what they're doing is taking components and replacing residues at the active site with new ones. Now, why are they tinkering with the active site? That's because that is where the work actually occurs. It doesn't make sense to change the outer structure because the outer structure does not affect catalytic activity. And so by changing out the amino acid residue at the active site, you can change the affinity for a particular substance, either increasing it or decreasing it. And if the affinity is greater, then that might mean that you can make a reaction occur more with that particular substance. That is one approach. Another approach has been to start from scratch, line up the particular amino acids, and make a totally new protein. Now, when you're doing that, the first part of it is to do computer modeling. And again, try and figure out which amino acid needs to be next to which amino acid, so that when I get my secondary and tertiary structure, I will then generate an active site that does the desired reaction. This is a lot of work. And if you have a look at this slide, you're not expected to memorize this, but it's an interesting one where industry is attempting to look at generating an enzyme that will serve to protect human health in an industrial environment. So thank you for your attention. That's it for now. Be sure to review, post questions, and enjoy. Thank you. Bye-bye.